Greetings. My name is Wesley Straub, Marketing Director here at Repligen, and it is my real pleasure to welcome you to this episode of the 2021 Repligen webinar series. As a note, before we get started, it's important for you to understand that this webinar is being recorded, and that is so that the content can be shared with others who weren't as fortunate to attend today's timing, right? So just a note on this platform that we are using today, we have a live question and answer panel off to the right there. You can just click at any time during the webinar to ask a question to our panelists. Those questions will be fielded at the end. So we'll take them, we'll receive them, and note them all throughout the webinar, but they'll be addressed at the end. If the question, you also have the option during that question period to simply write like follow up, like I'd like to answer to this question, but I'd like it on a separate phone call or an email. You can make that note in your questions as well, right? And then during the webinar, we have just two simple poll questions. The first one in the beginning here is just to see if we're all here for the same reason, and just the topic today is purification of COVID vaccines, and we're really just interested in making sure that we have a match there between your interests and what we're talking about. So we really appreciate if you run to the poll and answer that one simple poll question. We have one poll question at the end. We'll get to that at the end. All right. Other than that, the platform is really straightforward to use. The topic today is purification of COVID vaccines, specifically an affinity resin to purify vaccines. And a lot of affinity resins have been developed in this space, but it's really the timing around this one. This affinity resin for SARS-CoV-2 virus that is the real important part here. Repligen put an enormous amount of effort into developing this affinity resin really quickly as a response to the pandemic. So it's not just the product or the technology, I think it's the timing that makes today's webinar so important. And we have two people that are going to work together to deliver this presentation. First, we have Vikas Gupta, and Vikas Gupta is our Vice President of Downstream Purification and Gene Therapies, and he's really responsible for putting together the multiple technology, specifically Opus and the resin itself, into a solution to save you lots of time. And then we have Jamie Pizer as our Vice President of Research and Development, who's responsible for taking this idea of a resin from its earliest stages, developing the partnerships, putting the pieces together, getting it through development, then manufacturing, and now as a commercial offering to the market, so that we can really help the market develop vaccines just a little bit faster. So with that short and brief introduction, I am going to now hand it over to Vikas to get our presentation started. Thank you.
Thanks for the introduction, Wes. So Jamie and I will be walking you guys through this presentation. So I'll be first covering the whole product development efforts as to the partners that were involved and some product features and benefits. Then Jamie will be walking through the application data, and I'll finish off the webinar presentation talking about the integrated solutions or the formats in which we can offer this spike protein resin to the customers. So for those in the audience who might be less familiar with Repligen, so Repligen has been first to market in terms of offering a lot of innovative solutions to the bioprocessing industry, including Opus pre-packed columns, which obviate the need for self-packing of columns, as well as our filtration modules in both hollow fibers and flat sheet formats, and slope spectroscopy for process analytics, where you don't need to do any sample dilution for concentration measurements. So of course, this presentation is primarily focusing on NGL COVID-19 spike protein affinity resin. And as I said before, so this product is being developed in partnership with Navigo Proteins, which is a company based in Germany that helped us develop the ligand, and with our partners in UK, Purolite, that have provided the base bead, which has helped in the development of the resin itself. So we are all aware that a lot of vaccine manufacturers that are looking at spike protein subunits for their vaccine development, they're hoping that to elicit an immune response based on these protein subunits. So if you look at the graphic here, whether it's the receptor binding domain, the S1 protein, the S protein, or the entire spike protein trimer, this affinity resin should be a good binder for all of these potential targets. And as Jamie and the rest of the R&D team, we were working with a lot of customers since October of 2020. One of the common themes we were hearing from a lot of customers was that they found the use of existing affinity resins very complex to use. The purification process entailed multiple chromatography steps. They were dealing with lower yields, and all of this was impacting their development timelines. And as you can imagine, with this pandemic raging across the world, every day matters in terms of accelerating the timeline. So based on the feedback we have received, this affinity resin will help our customers accelerate the development process significantly. So back in March of last year, when we had just started hearing about this pandemic reaching Europe as well as the US, Navigo and Repligen, we sat together to figure out how we could get involved in the vaccine and drug development process. And Navigo worked at an unprecedented pace and developed a ligand, a target ligand by May of 2020. And then it was tested on various substrates to start providing test samples, by July of 2020. And since then, we have gone through the prototype and resin scale-up work, which finished in December 2020, and we will be providing validated and GMP samples by April of 2021. And if you look at the two companies here, Navigo with their expertise in protein engineering and Repligen's history of being the largest producer of protein A ligands, and with our resin immobilization expertise, I think the two companies are very well placed to provide this resin to the market, backed up by a very robust supply chain. And looking at some of the high-level product specifications, so as I said before, the resin primarily targets RBD or other spike protein subunits that do contain the RBD. It yields greater than 90% purity, and based on some of the customer testimonials, we are getting up to 20 grams per liter of DBC. And now I'll hand it over to Jamie to walk through some of the application data.
Thank you, Vikas. So I'm going to walk you through some of the specifications that we set out for the resin, as well as some applications data that we have generated both with internal material and some antidotes from vaccine developers. So on this slide you see here, this is our target product profile and some of the results that we put in place as specifications for the final resin. So you see here the binding target, of course, it's RBD from the SARS-CoV-2 spike protein. We're getting capacities of 20 grams per liter or better for the receptor binding domain. Important feature is that the ligand is an engineered recombinant protein affinity ligand that is expressed in E. coli, much like our protein A products that we manufacture and sell. The bead itself is on a cross-linked agarose of 85 micron average particle size. This is a commonly used support within monoclonal antibody manufacturing and is well suited for these RBD proteins as well as larger molecules as well. Lucian conditions, we're able to elute RBD and other spike proteins at around pH 3.5 and a little bit higher. Also, CIP can be done with common solutions that are known to industry, like 0.1 normal sodium hydroxide. So in this slide, you'll see some performance results. So in general, the dynamic binding capacity has been shown to be greater than 20 mg per mil for the receptor binding domain. The graph above shows dynamic binding capacity versus the residence time or loading flow rate. And as you can see, even at short residence times, we're getting very high capacities. Another important feature is the base stability. So with 0.1 normal hydroxide, exposure of 20 hours, we're still retaining approximately 90% of the original binding capacity. The acid stability is also excellent, and we see no decrease in performance throughout 50-hour exposure. So now I'm going to jump into some applications data. First, I'm going to talk a little bit about the early evaluations that we started back in the September, October timeframe. And this was done with multiple leading vaccine manufacturers or developers. They had a diverse range of expression systems and a lot of different spike protein variants. Ranging from RBD all the way to Filtrimer and also larger molecules like trimer oligomers, so dimers or trimers of trimers, so very large molecules. And in all of their evaluations, it came pretty quickly and clear that the affinity resin was performing at very, very high levels, meaning they were able to eliminate so several chromatography steps that were in their original processes. They were able to reduce down to one affinity step and maybe one polishing step, and also increase the purity from what they were getting with original processes from sometimes lower than 85% to well over 90-95%. It was also shown that the purity from this specific affinity resin was much better than seen in some tag technologies. So a great benefit there. Also increased yield. So if you have several unit operations that you need to run and you can eliminate those, of course, you're going to get high improvements in process yields. Another point that was brought up or features that with original processes, a lot of the HCPs are problematic HCPs that just could not be cleared with traditional ion exchangers or tag technologies were able to be removed with this specific affinity resin. And also the final product is confirmed to be still active in the ACE2 receptor binding, which is an important feature. Next. So I'm going to get into a little bit of data from purification from different expression systems. In this case, it was a receptor binding domain expressed in XB293 cells. The loading solution here was a somewhat dilute feed, I believe 0.2 to 0.5 mg per mil, loaded, washed, and then eluted at with a pH gradient of pH 6 down to pH 2 at 15 column volume. So as you see from the chromatogram, the pH peak of elution was 3.5, and from the gel that we got single, very high purity, single band resolution within the gel. Now I'm going to talk about another purification that was done with CHO cells. So RBD, again, expressed in a CHO cell culture. This experiment was done with a again, a dilute feed of 0.2 grams per liter RBD, loaded to approximately 80% of the resin capacity, which in this case was 17.3 mg per mil, and then washed with a series of wash buffers. So first PBS in high salt condition, and then back to PBS, and then eluted with acetic acid, pH 3.5. As you can see from the chromatogram, the elution comes out in one very sharp single peak, and then you can see the small CIP peak afterwards from the cleaning. And also on this slide, we show that the elution fraction itself contains good ACE2 binding, so it retains its activity following the purification and elution at pH 3.5. To follow up on this experiment, other analytical methods were run, so SDS Page and size exclusion. You can see here from the SDS Page gel, the material as loaded is very dilute, as seen in lane 2 at approximately 36 kD. And in lane 3, we see the flow through with some absence of that band, and also the wash in fraction 4 and fraction 5 show all of the HCPs washing from the column, and then the elution fractions in lane 6 and 7 loaded at 3 micrograms and 5 micrograms. These elution fractions show a very distinct single band of very high purity, and also what we see is high yield, greater than 85%, and great clearance in HCP, so a 2.95 log reduction using a CHO HCP commercial kit. Also, we confirmed, like I said in the last slide, the ACE2 binding activity in the elution fraction, and there was no ACE2 activity found in any of the flow-through or wash steps. As you see the size exclusion chromatogram, the elution is also very pure and contains both RBD dimer and monomer. This RBD dimer formation is from the cell culture itself and not induced during the elution part of the protocol. So lastly, I'm going to follow up with a study that we did to look at the lifetime of the resin. So in this study, we did 50 cycles with a modified protocol based on the last study with a more concentrated feed. So the feed was at 2 grams per liter in this case, and the CIP was done with 0.05 molar sodium hydroxide each cycle, except for every 5th cycle, CIP was done with 0.1 molar sodium hydroxide. So a little bit stronger cleaning every 5th cycle. We also had benzyl alcohol in the CIP solution to maintain a more bacteriostatic or a cleaner environment. Since this study ran for a long time, we wanted to keep the system nice and clean. As you see here in cycle 1, the load, washes, and elution being very nice, clean, sharp elution peak. And in slide 2, you'll see here cycles 1, cycles 5, and then all cycles overlaid in the chromatograms. And the difference between cycle 1 and cycle 5 is within the CIP. So you'll see a second peak after the elution peak. It's a little bit larger in cycle 5 due to the stronger CIP condition. But overall, all the cycles, when overlaid, you see nice consistency in elution. And if you look at the results for yield, the yields were all greater than 85% throughout the 50 cycles. Dynamic binding capacity after 50 cycles was still greater than 88%. So we maintain the resin capacity throughout the study. and also the HCP reduction was at a high level of 98% throughout the entire study. And that concludes my section.
Yeah, thanks for the technical update, Jamie. So if we look at the formats in which we can deliver this resin to our customers, of course, you are well aware of the entire range of Opus pre-packed columns. The resin samples are available in 1 milliliter and 5 milliliter Opus minichrome columns for some early evaluation work or process development work, and you can scale up all the way up to 80 centimeter columns as well. And of course, I mean, if you are interested in just buying loose resin, we can make that available and actually deliver fully integrated solutions via these chromatography skids with fully integrated single-use flow paths. So we are very excited in bringing this resin to the market, and we hope that it will help our customers in their vaccine development process. Feel free to ask Jamie or myself any questions right now, or send us an email. Or in case you have any business related questions, you can reach out to your local sales representative. Thank you.
Okay, Vikas. So we're going to open up the floor to some questions and answers. We've had a couple questions come in during the presentation. I'll read off the first one that came in. We're just going to take them chronologically. First one that came in this is to Vikas and Jamie. Will the resin work on newly emerging variants?
Sure. So this work is ongoing. We've had some success so far with the limited amount of materials that we've been provided. So with the British variant, we've confirmed activity with that, and we're waiting for some of the other variants to come in so we can confirm testing as well, or confirm binding.
Okay. Second question that came in, will the resin purify whole virus? Going back to the beginning.
So that's something that we have limited information on. The resin was intended or developed with the intent of purifying protein-based vaccines. So whole virus being in a lot larger molecule or virus is something that we do not, again, have a lot of experience with. We're looking for some feedback from customers.
Okay. Third question, is the resin ready for GMP manufacturing?
Sure, I can answer that. So we've completed our validation testing, and now we're in the final stages to make it available for sale, and this would be for GMP manufacturing use.
So we're targeting like third week of April to start delivering GMP ready resin.
Okay. Next question, I think, as mentioned in the beginning, it's all about time. Next question about time. What is the lead time for 45, 60, 80 centimeter column? I'm going to assume that means a 45, 60, 80 packed with the SARS-CoV-2 resin.
Yeah, so the current lead times for 45 and 60 centimeter columns is 6 weeks, and for 80 centimeter columns is 8 weeks.
Yeah, 6 8 weeks. Skipping over to the ligand, we have actually two questions around ligands. So is there any info on release of the ligand, during assessment of resin performance after NaOH and acid exposure?
Yeah, so maybe that ties into a question about how to detect leach ligand. So we're also developing and will provide a leached ligand ELISA kit. This ligand has a lot of homology to normal protein A, and therefore we can use our base protein A ELISA kit for detection. So far, what we've seen from trials, the sodium hydroxide exposure will eventually degrade a protein-based ligand, but our ligand is pretty sturdy and holds up well for long-term exposure with sodium hydroxide.
Okay. So Jamie, I'm just going to couple that one with one that I'm going a little bit out of sequence. There is one in there for an ELISA kit for leaching assay to detect the ligand when it comes off the resin.
Yes. Yeah, that will be available.
All right. There's a detailed question on basically how the lifetime study was run. Basically, did you perform a blank carryover during testing, during the lifetime study when you executed that study?
No, that was not performed in that detail. We're currently doing more lifetime testing, but yeah, that's a good question.
Okay. All right. Person's asking for which post-load wash, basically buffer, is best for HCP clearance. Any suggestions around that?
So there can be a lot of optimization around that. Just with a standard PBS, phosphate buffered saline, we see reasonably good clearance. Additions, other additives. I know a lot of companies have their own proprietary washes that they use with protein A based processes. These are also applicable. So happy to discuss on a case-by-case basis.
Okay. Just to try and keep us on the CIP steps, I'm going to go with has a half molar sodium hydroxide exposure been tested on the resin for stability?
Yes. Yeah. So that has been tested. It holds up. I'd say it holds up well to 0.5 normal sodium hydroxide, but again, as a protein-based ligand, it will degrade its performance over time. So again, happy to discuss that further.
All right. I have two similar questions from two different attendees. The first one is basically biological activity of the spike protein. Is it impacted by the purification process? But we have another one. Just hold on for a second. Have you compared ACE binding with RBD purified using the resin and other types of resins? Basically, I think both questions are asking, what does binding look like after elution?
Right. So internally, we have not done a head-to-head comparison of RBD purified with other technologies. Externally, I can say that we've had several tests and head-to-head comparisons with results.
I don't know if people can hear Jamie.
Sorry, Vikas?
Go ahead. I thought there was a breakdown. Yeah, go ahead, please.
Yeah, I was going to say that this is not with just RBD, but also full trimer and other subunits as well.
Okay. All right, so I think that takes care of basically biological activity of the spike protein impacted by purification process. And then we have what is the typical binding and elution condition kind of coupled in with activity after?
Sure. So it's very similar to antibody processes with protein A. So neutral pH condition for binding or load, and acidic condition around 3.5 for elution. We do have options to increase the pH if that's a concern. But generally, an acidic environment is the best condition for elution.
Okay. And then from Clara, if I understand well, you need a second chromatography step, cation or anion, right, to increase purity after the affinity step. Any suggestions on whether a second step is required?
Yeah, I mean, I believe that even so, with monoclonal antibodies or any molecules that are purified with affinity chromatography, a polishing step is required to remove any leftover small traces of HCP, DNA, and also potential for the leached ligand itself from the affinity step. So, yeah, it would be recommended to do ion
And if,
exchange or multimodal step following affinity step. Yep.
and if I may add there, Wes, so
Mhm.
we did an extensive customer VOC, and most customers who are using legacy processes, they have 3 to 5 chromatography steps, and even other complex unit operations, including precipitation. And the experience they have had with our resin is they've been able to cut down the number of steps down to 2 steps, one affinity and one polish, like Jamie just said. And the yield improvement, back into the 90% range. And I mean, the purity and the yield is, up 3x to 5x for certain customers. So we are really, really happy to see the advantages this resin is offering to our customers.
Okay. Nice. Okay, that's all we've got. We have a few other questions that are kind of more appropriate for detailed follow-up offline. So no matter whatever question you wrote in the questions there in the inbox, we will follow up with you on those. So that wraps up our Q&A section. Just as a reminder, this webinar was recorded. It will be posted live. It will be posted on the Repligen website for share. So if you have a colleague that you want to share it with, that's definitely possible. And we will follow up with that link at the conclusion of the webinar. So thank you everyone for attending, and we look forward to seeing you on the next episode of the Repligen 2021 webinar series. Thanks.
Thank you.